I am now ready to extract that DNA from our PCR products. And I'm going to do that using a PCR purification kit. Now, the one that is in your lab handout, we actually don't have all of the ingredients for that. So I am going to use a PCR purification kit that I have in my lab that is very similar. It still uses these spin columns. The sample gets loaded into these columns that are then going to go into a centrifuge to be spun down. So we will start this out by using something called Buffer PX that is in this kit. This is from a company called Epoch. It's a little different from the kit that was in your lab. So what this kit asked me to do is to use 500 microliters, so a half of a milliliter, so now I'm using the P1000, which I haven't used yet. And we have these blue tips for this, and I'm going to add 500 microliters of this into 2 new tubes. All right. And then I'm going to take our PCR products that are still in our little tubes here, and I'm going to add them to each of those tubes. Now, make sure that you label things so that you know which one is which. Here to label. I'm going to vortex those samples. So we've seen how to use this device called a vortexer before. That mixes everything up. All right. Okay, now that these are mixed, they're ready to load onto these spin columns. So this is actually a column that is stuck inside of another tube. So that column has a filter in it, and that sample will be applied to this top column, and then upon centrifugation, flow through. But the DNA will not. It will actually bind to the column. I'll go load all of that sample, and I have those columns already labeled for me. I put it all on there. Right, now we have the sample in the tubes or in the column, and I can put these in the centrifuge. Now, it's really important with these centrifuges, and I think you can see it here, that, move this a little closer, that you have those samples across from one another so that this is balanced. And there are these lids. You'll know if it's either not balanced or doesn't have the lid because it will sound awful. So I only need to spin this down for about 30 seconds at low speed. So I'm going to turn this down. All right, let that go for 30 seconds. 30 seconds have passed. I'm going to take the samples out of the centrifuge, and you can see now, oops, that the liquid is all at the bottom. It's no longer in the top of the spin column. That is waste. Our DNA is stuck in the filter of that column, so we want to get rid of that liquid that's in there. All right, so now we have these empty spin columns. We're going to wash the spin column with 2 different buffers. Each of them are going to have a concentration of ethanol in there that will be used to essentially wash those salts away and allow for better binding of that DNA. Add that into the first one, and I'm being careful not to actually put my pipette tip into the column, or else I would need to exchange that pipette tip and change it out each time. If you're ever unsure if you've touched something, it's always best to just change the pipette tip And we will spin this for 30 more seconds. Okay, the centrifuge is done. Take our samples out. Again, this is going to be waste. We're going to discard that waste. And wash one more time. This kit has 2 different washes. It calls the first one WN, and then the second one is WS. So we've done WN, and now I'm going to add WS to each of them. And spin this one for another 30 seconds. Start. Centrifuge is done. One last time that we are going to discard what is in here. And now we spin this one more time in those collection columns. That's what these things are called. That's the other tubes that they're in. One more time in here. And this one is going to be for a little bit longer. So I'm going to spin this now for 2 minutes. And what that will do is allow all of that ethanol that's in there to either evaporate or spin into the column, so that that is now washed DNA with no ethanol associated with it, so that we can elute it in the next step. So now a 2-minute spin. Those collection tubes do have some liquid in there, so that means we needed that longer spin and that faster spin. I didn't let you know that, but that was actually a faster spin that we did this time. And now I'm going to take those collection tubes and toss them, but I want the columns. So these columns are going to go in brand new, labeled, empty, sterile centrifuge tubes. And they're going into the tube that's associated with a label that corresponds to the column, the right DNA sample in each. Now we want to add our elution reagent. This can either be elution buffer, which comes with many of these kits. This is stabilized so that it doesn't change pH for that sample that you're using, or you can just use water. So I'm going to use sterile water. And I'm going to carefully pipette that water onto the membrane, but without touching the membrane. So you don't want to pierce that at all. I tap it down there, make sure all the liquid is in the tube, or is on the filter. And we're going to let that sit for a few minutes. I'm going to do the next one. And now we wait. The protocol says 2 to 4 minutes to let that water or elution buffer soak in that DNA to elute it off the column after we spin this. We'll let this sit for 2 minutes. Our 2 minutes are up, so we're ready to centrifuge this. Again, balance them. And then I'm going to spin for 1 minute, and then I'm actually going to apply that same sample to the column again and spin it one more time. So our 1 minute spin has completed, and I will show you what I do. Now, this is an optional step, but it tends to allow us to make sure that we have a good yield. So the water that we spun down is now at the bottom, and that should have our DNA in it, but it's possible that there's still some DNA that wasn't properly eluted. So I'm going to reapply that same sample by cutting it from the tube and putting it back in the column. Do this for both of them. Get it out. Okay, putting it into the column. You know my hand's in the way, you can't see. There we go. And we will spin this for 2 minutes, and that should be enough to get all of that sample down into the tube so that we can record or measure and then record the concentration of that DNA. So 2 more minutes and then spec it. All right, our samples are now out of the centrifuge. So those columns that we have, our DNA should all be collected down in this tube, so we can get rid of them. Throw those away. My tubes are labeled, so we know which sample is which. And now these are ready to nanodrop. I am not going to make a video of nanodrop because you have already done this in lab, but I will let you guys know what the concentrations of those samples will be and then how they will be prepped in order to sequence them.